Well, thank you, Andrew, for a very warm and generous introduction there. Very restrained as well, going by your previous. I want to thank the committee for inviting me. I'm very honoured to be giving this talk. And yeah, I'll see what I can get away with saying today. Before I pile in, I want to acknowledge and thank all these wonderful co-authors here. Just working with these brilliant people has really helped me to cover a broad range of topics, and it's been really enjoyable. On the left-hand side of the screen, we've got the sort of genomics, experimental evolution people. So Rob Young and Sinead Collins, Boris Sauteré, who I've just seen is here, and David Clarkson have helped me working with adaptive dynamics models of plankton ecosystems. Steph Dukowitz and Kale from, Steph's still at MIT and Kale previously at MIT and now at Southampton and NOC, working on the sort of biogeochemical modelling and biogeochemistry side of things. Similarly with Jamie Wilson and the people on the right-hand side, Andy Ridgewell and Samantha Gibbs, have helped extend some of these ideas into Earth system and paleo perspectives. So it's been really wonderful working with these people, and this is the only way I've managed to cover this sort of broad range of topics and scales. So what I want to talk today, as suggested by the title, when everything is not everywhere but species evolve, is looking at three common assumptions that I've used a lot and a lot of other people have used in trait-based ecology. First of all, being that population traits are fixed. So often assuming that species and populations can't evolve. And then this assumption that everything is everywhere, but the environment selects. I've broken that up into two parts, and you'll see why later in the talk. But these sort of underpinning assumptions of a lot of what we do in this area. So in my talk today, I'm going to give a brief introduction to ecosystem function and biodiversity, why we care. I'm going to talk about some trait-based approaches to dynamic ecosystem modeling, so specifically simulation models based on differential equations rather than empirical approaches, and then talk about these fundamental assumptions. I'm going to look at these assumptions from three projects, looking at the evolutionary recovery of a microbial food web after the end Cretaceous mass extinction. I'm going to look at constraints on global plankton dispersal using a population model in a global ocean. And then finally look at some unpublished work looking at how rapid evolution of microbes can lead to a kind of emergent neutrality and coexistence. And then end with some conclusions on what we can learn from this. So looking at marine ecosystem function and biodiversity, we obviously have these very important ecosystem services, including carbon sequestration and food webs. And one of the reasons I like working with dynamic models as opposed to empirical models certainly very, very valuable. But I'm really interested in these feedbacks between environment and an ecology. And this was summarized really nicely in this early paper by Redfield, and he said it's a recognized principle of ecology that the interactions of organisms and the environment are reciprocal. And we see here how the growth of communities impacts the environment, and then those communities, the environment regulates the growth of ecological communities, but the growth of those communities then feeds back and affects the environment. So we have this feedback loop where you can't really unpick these two effects, and dynamic models, ecosystem models, are a really good way of looking at that. And when we think about function of plankton communities, it's a very important way of thinking about this is these functional groups. We have, small picophytoplankton associated with the microbial loop. We've got diatoms and other silicifiers associated with the silica cycle, but also with export. Nitrogen fixers, all these different functional groups play these different functional roles, and it's important to get these effects into models. And this very fundamental idea is key to the development of plankton functional type models, where they put these functional groups into a simulated food web and evaluate that in an ocean environment model. These models are parameterized based on laboratory experiments. So looking at how fast diatoms versus Prochlorococcus can grow in a lab, and then you put that into the model. The trouble with this kind of model is it does require hundreds of parameters to get a model of this complexity when you're specifying each individual component that you think is important. And at the same time, these functional groups actually mask an enormous amount of diversity within those functional groups. So the diatoms are an extremely diverse group with extremely diverse traits. So one way of thinking about this as an alternative is to look at trait-based approaches. And this is a nice summary by Elena Lichman et al. Looking at these key traits and treating these traits as ideally as continuously varying populations of the phytoplankton, and try to represent those key traits rather than specifying the individual components that you put into the model. And so when you do that, you can say, well, we've got a load of phytoplankton of different sizes, but we know how their various traits, like growth rate, metabolic rate in this instance, vary with size. And this gives us a much more constrained and low parameter way of building that complexity into the model. And we can also look at things like temperature dependency as well, and see how once you put these sort of more general constraints into the model, you can then look at the emergent properties. And this is this is what sort of defines the Darwin modeling approach, where you have a load of prior constraints, and then you randomly or not randomly generate a load of candidate populations to put into your environmental model. You run that model forward and you let fitness and selection pick out the best populations, phenotypes in different environments. And this is a great sort of setup for modeling the complexity of marine ecosystems and those feedbacks. And this is the classic example from the Follows et al. 2007 science paper, where these randomly generated types ended up growing in environments where you'd expected them. So all this diversity was generated with a much lower parameter complexity than that original functional type modeling approach. So as I said, I want to look at some of the assumptions of these models. And again, as I said, these assumptions are firstly, models like this often, but not always, will assume that population traits are fixed. So once a population is in the model, it doesn't change its growth rate, it doesn't change its grazing parameters or whatever. Again, often, but not always, will assume that everything is everywhere. So it's very easy for populations to disperse in the model. There's not much local extinction going on. So things can spread out quite well all over the globe. And then the environment will select the best species or population to grow in that particular environment. And that's how they work. What I want to do in this talk is look at this part of the, so I'll just go back one step. So we've got this feedback loop that Redfield alluded to. So we've got the environment impacting the ecology, and then the ecology feeding back on the environment. What's missing is a second feedback loop where we've got limits to dispersal. We've got evolutionary adaptation of organisms feeding back to change the fundamental traits of the populations in the organism. And that's what I want to talk about today, is how these evolutionary and dispersal related feedbacks can impact the outcome of marine ecosystem models. So I'm going to talk about that through 3 different case studies. The first, I'm going to look at population traits being fixed or evolving. I'm going to look at a case study where we have evolutionary recovery of a post-extinction food web 66 million years ago. The second case study is going to look at how temperature-based selection can limit the dispersal of phytoplankton, perhaps questioning this assumption that everything is everywhere. And finally, I'm going to use an individual-based model to show how rapid evolution can lead to a kind of emergent neutrality and a lot more coexistence than you might expect, given the competitive exclusion principle. Okay, so the first case study is a paper led by Sam Gibbs, published in 2020. And what we showed was there was an evolutionary shift to mixotrophy among coccolithophoric nanoplankton. And that was key to rebuilding a functional food web in the aftermath of the end Cretaceous mass extinction, when a lot of the large phytoplankton in the community were wiped out. So Sam and her colleagues looked at nanofossil data and found some really interesting patterns. Excuse me. And then we use an evolutionary ecosystem model to explore those patterns. So the starting point being the data, we've got nanofossil data for this period from 66 to 64 million years ago, and a bit into the nearer time as well.
And you can see how after the mass extinction event occurring 66 million years ago, this horizontal line, we see basically a succession of different groups over millions of years, ones coming in and going out and being replaced by others. And when you look at those particular fossils in detail, you can see among these coccolithal species, there's some really interesting openings here in the shells. These are extremely well-preserved fossils, and a lot of them appear to have these openings. And when you look at modern coccolithals over here on the right-hand side, we can see the similar structures. And this appears to be related to, this is effectively a flagellar opening. And this is something that allows these organisms to be motile and also possibly to ingest prey. And motility is important because to get hold of prey, especially get hold of non-motile prey, you need to be motile yourself. So we took this as evidence of these photosynthetic organisms were also ingesting prey, so they're mixotrophic. If you then look again at this fossil record, we can color that by mixotrophic species in blue and strictly autotrophic species in green. And we can see it after the mass extinction event. There's a lot of the survivor species on the left are mixotrophic, and there's also some new mixotrophic species coming in for a brief period, newly emerging in these fossil records. And when we extend that over a longer period from 66 to 62 million years ago and look at it in a bit more detail, you see this interesting pattern of sort of the lead authors called it acme successions. So you see these species coming in and rising to high abundance and then declining again to be replaced by another species. And a lot of these acme species are mixotrophic nanofossils, nanoflagellates. So this is the interesting pattern. So we tried to look at this using a very simple trait-based model. And this is effectively the model structure here. We've got in blue a nutrient being taken up by green phytoplankton. And then we've got a grey mixotroph group that takes up nutrients and ingests smaller prey. And then we've got the pink zooplankton. And we model these all using a generic equation. So we have the population growth rate set as a function of nutrients, and this is just a Monod function. And it also increases by ingestion of prey, and it decreases by being preyed upon by predators, by linear mortality and a quadratic mortality. And to add a bit more complexity, we set up a system where you can have a whole different range of sizes and a whole different range of trophic strategies. So in the model, we had about 51 size classes and about 51 trophic strategies as well. So from small being about 0.5 microns to about a few millimeters maximum size. And on the left, we've got strict autotrophs. On the right, we've got strict heterotrophs. And in the middle, we've got degrees of mixotrophy. And in this everything is everywhere type modeling, we just seeded the model with a small number of all of these different trait combinations and let it run forward. And then we looked at an alternative approach where we allowed an evolutionary emergence of different types rather than saying everything is everywhere. So the advantage of this trait-based approach is you can get sort of a nice reliable parameterization. And this data is drawn from Kyle Edwards' work, Emilio Marion's work. And so we have this size-dependent maximum growth rate that peaks in intermediate sizes and with the smallest and largest phytoplankton growing a bit slower. We have size-dependent nutrient affinities, so the smallest phytoplankton have the highest nutrient affinities, and smallest phytoplankton also have the highest grazing rates. And we also have on the right-hand side this data showing predator volume versus prey volume, showing that amongst these unicellular grazers, it's generally a 10-to-1 ratio as the most common predator to prey ratio. So we also included that in the model.
in terms of a trophic trade-off, we needed to have a way that the mixed traits weren't were slightly not as good as a strict autotrophic traits and not as good as a strict heterotrophic traits. So we just have this linear trade-off. And I've never really seen any evidence to support this, but it seemed kind of intuitive. And yesterday, Kyle came up with an amazing analysis showing that perhaps this trade-off is actually there, and it's some sort of empirical grounding for that. So it's great to see that yesterday. So with these trade-offs, the size-based and trophic trade-offs, we can run the model forward. And first, we look at this this everything is everywhere simulation. Jorn Bruggeman called this a system of infinite diversity. So all trait combinations are possible. And if the habitat is right for that any particular trait combination, it's going to survive and grow. So if we see the model with this low seed biomass across the trait space, within 20, 50 years, certainly with 100 years, we get the emergence of a stable community dominated by phytoplankton and zooplankton, with a few mixed trophic species in between. But it's a very quick emergence, and we don't see any of this sort of successional patterns that were shown in the data. So instead, we can drop this assumption and switch to a model where actually everything isn't everywhere. And we're going to seed the model just with strict phytoplankton below 2 microns in diameter. And if we're going to get zooplankton in the model or mixed traits in the model, they have to evolve from this seed community. And the way they evolve is by producing mutants. So a growing population will reproduce itself mostly with exactly the same traits, but some of its descendants will have slightly larger size, some will have slightly smaller size, some will be more or less mixotrophic. And those mutant offspring can outcompete their parents effectively, and that's how we get evolution in the model. So we run this model forward. Initially, we have a bloom of phytoplankton, but then we start to see mixotrophs coming into the system, and we see this gradual diversification of the system. And this video is running twice as fast as the first one. We've already had 350 years. So over a much longer relative timescales, we see the emergence of this food web structure. We're also seeing mixotrophs being a much more important part of the system, and this sort of evolutionary transient. And I won't run this all the way through. What I'll do now is switch to the same simulation, but now we've added this surface at the top, and this is effectively the fitness landscape. And this is the specific growth rate of each trait combination, whether it's there in the model or not, given the current environmental conditions. So this is you can see here when we seed the model, there's a big peak in the intermediate sized autotrophs. And that's to do with these organisms. This is a high nutrient initial condition. So these autotrophs, the intermediate sized ones, can grow the fastest, so they have the highest invasion fitness. And then we see it tailing off as they become more the more heterotrophic side of the trait space. And this is to do with this linear trade-off. As we run that model forward, I'll just pause it there. We've got this initial phytoplankton community, but there's no grazers in the model because they haven't had a chance to evolve yet. And what we see is this massive peak in the fitness landscape in that part of the trait space where you have active grazing on this initial. So this is like associated with the size class is 10 times larger than this initial population. So you see this massive peak in the fitness landscape. This is effectively an unoccupied niche, but the system has to evolve towards that, and that takes time. So they can't suddenly jump there. They just have to move step by step. Oh, beg your pardon. So when we run that forward, you'll see that happening. And as they do move into that niche, the fitness landscape becomes flatter and flatter. And also you'll notice that the community always exists sort of around the intersect with this grey plane, which is the zero invasion fitness. And this is because these systems are at an ecological equilibrium. Their net growth rate is very close to zero, but over evolutionary time, they're climbing that fitness landscape and modifying their environment so they eventually reach an evolutionary equilibrium as well. So when we return to the data, we can see this successional pattern in the fossil record. We also see that successional pattern in the model where one species has been replaced by another. And what's happening there is that evolutionary replacement of species. One comes in, goes to an ecological equilibrium, produces mutants, and then that one outcompetes the original one. And we see this evolutionary succession going forward. So the model is quite nice to work with because it applies modern ecological parameterizations, but over evolutionary timescales. When we run that model with the everything is everywhere assumption, the model recovers too fast. The evolutionary model also recovers way too fast. We're talking about a million years in the fossil record, 1 to 2 million years, and it takes about 1,000 to 2,000 years in the model to run. But at least it has, the timescales are all wrong, but this is because we're kind of using a microevolutionary model to look at much slower macroevolutionary processes effectively. And this mechanistic model helps to interpret the paleo data. So the second case study, selection versus adaptation in dispersing plankton. And what we found was that environmental selection seems to place strong constraints on global dispersal. And even when we allowed those populations to evolve extremely fast, it wasn't possible for organisms to spread rapidly around the world because they had to cross high thermal gradients. And to do this, we used a global, that says metagenomic, that should say it's global metagenomic data with a metapopulation model. So this is really getting at the everything is everywhere assumption. The question is sort of, if you think about how many plankton there are, the best estimate is there's around 1 billion plankton. And as you can see here, that's more than stars in the universe. This is an extremely high number. And the implication of this is perhaps that because these organisms are so abundant, it's very easy for them to disperse globally. So there's never an environment where an organism can't reach. And this is underpinning the everything is everywhere, but the environment selects. So why is this assumed high dispersal important? Well, if we assume that everything is really everywhere, we can't have allopatric speciation and endemic species in microbes. We can't have species being geographically separated, allowing them to evolve into new species. The idea of microbial invasive species doesn't really make any sense either. And we can think about how it affects the adaptive potential of a community as well. Andrew asked Adam the question yesterday, you have this sort of reciprocal inoculation of a community. That wouldn't make any difference under this assumption because the community in both areas is both going to contain the same organisms, and it's really the environment selecting it. So that doesn't have any effect. But if everything isn't everywhere, we get a very different result. And the other key point is, can populations track shifting isotherms under global warming? If everything is everywhere, it becomes much easier for them to do that. So Tom Fenchel argued that everything was really everywhere, and this means that habitat properties alone are needed to explain the presence of a given microbe, and historical factors therefore become irrelevant. So we can look at this in modelling studies. Ferdy Hellweger's 2014 paper looked at this, and they used a individual-based model. And they effectively set a carrying capacity in different parts of the ocean and looked at how quickly species were dispersing. And they showed that in this model, the rate of global dispersal was not fast enough to break down the local effects of diversification. So you get this emergence of biogeographic regions. So there is a degree of endemism here in this model because it's dispersal limited. So they conclude that neutral evolution with dispersal limitation can produce these biogeographic patterns. On the other hand, Johnson and Watson used a Lagrangian particle tracking model, and this is just neutral traces again, with no interaction with the environment, no ecological processes, to see how quickly ocean regions could be connected. And they developed this matrix of connection times in the ocean. And they found that the median connection time was on average less than a decade. So this is extremely fast. And this would imply that, actually it's much shorter timescales than are needed for some sort of biogeographic structure to emerge. So perhaps everything is really everywhere. The trouble with these, first of all, these models completely kind of disagree with each other on the question, but they also ignore any selection. The Johnson and Watson model ignores the possibilities of local extinctions. They're treating plankton just like passive traces that can disperse without limits. And the Hellweger model ignores, doesn't ignore population sizes, but it assumes very low population sizes of about 100,000. And this really impacts the level of diversity a community can maintain. And both of these models, being neutral, ignore the role of adaptation as well. So we can also look at the data. This is a study based on Tara data. And they looked at the Bray-Curtis similarity between communities within the Tara oceans, and they related that. So they have metagenomic similarity based on this metric here, and they clustered the organisms based into different clusters based on community similarity, and showed that there were these sort of biogeographically similar regions. And they also showed that this similarity decays the further you go in terms of travel time in the ocean. So this is implying there is some distance-based effect where dissimilarity emerges the further you go away from an organism. On the other hand, again, using the Tara data, and Enrico Sergiacomi's paper presented this species abundance distribution, where you have a lot of extremely rare, possibly maladapted species in the environment. And these have a sort of power law relationship. But the key thing is you have a few locally dominant species that sit on a different curve. So this black line is sort of like the rare biosphere background community of highly dispersed organisms. So that's the everything is everywhere. And then the red points are the environment selecting for the locally well-adapted species, and they're becoming locally dominant. So again, the models disagree, the observations disagree. So I thought I'd do a study to add some more disagreement and not clarify anything. But we can look at the different mechanisms in a bit more detail. So in our paper, we asked the question, how rapidly can a local population disperse globally when you account, well, you can ignore selection and assume a neutral model. You can account for selection, but not evolution, or you can add in adaptation and evolution and see how this affects the ability of a local population to disperse globally. The model accounts for very high population sizes. So it's a population genetic model that correctly accounts for, or not correctly, but has a better representation of stochastic demography, local extinctions, and that sort of thing. It can include natural selection, it can include evolution, and it includes a representation of the ocean transport. So that representation of the ocean transport is based on a transport matrix. And we do that here with two locations, the two boxes. And in those two locations, we've got a blue population and a red population. We can represent that as a matrix with the locations being the two columns and the populations being the two rows. So in the first location, we're completely blue, and in the second location, we're completely red. And you can mix that using a transport matrix T. And this just takes a fraction of one box and puts it in another box and mixes up. And this is how we represent the ocean circulation. And then the population genetics or stochastic demography part is, once you've mixed them according to this transport matrix, for these two populations in each location, we can then draw the abundance at the next time step based on the population frequencies in each location you draw from a binomial distribution. And for high abundances, this is exactly the same as just mixing it together. It's very deterministic. But the key thing is that low population abundances, things actually have a chance of going extinct because they've been drawn from this distribution. So in the more complex model, exactly the same form of the equations. We have the original distribution X, and then we mix it by the transport matrix. And then at each time step, we draw from the binomial distribution to work out what the abundance of each type at the next generation is. And so in this simulation, we seeded one location in the Weddell Sea. Is that the Weddell Sea or the Ross Sea? I don't know. In the Southern Ocean, and looked at how quickly it would spread around the world, just as a passive tracer. And this movie I'm about to show, it doesn't have the time in it, but the whole movie is about 30 years of model time. So you can see that actually very rapidly that population spreads around the world, and all points are connected. So that's the principle of the model. And then the question we can ask is, if we take a load of different sites, these are 96 different seed populations, and look at the time it takes for those to become connected. And initially, when we run that in the neutral model, we can see that 95% of these points are connected within 10 years. And this aligns with Johnson and Watson's estimate. Which is suggesting that everything is everywhere. But when we think about it, for a species to go from the North Atlantic subpolar gyre here to the Southern Ocean, it has to cross a very different set of environmental conditions. It has to cross the equator, where waters are going to be 30 odd degrees warmer. And it has to, as it's doing so, it has to compete with species that are already adapted to those warm waters. So it's actually a real challenge for them to do that. So the way we represented that in the model was just to put this selection coefficient where species have an optimum temperature. And if the environment deviates from that optimum temperature, they have a lower chance of being selected in the next generation. So each species at each location is given the thermal optimum of its initial location. So this one would start off with, say, around 0. And it's going to have to, without evolution, it's going to have to compete with species that are much, much better adapted to their local environment than it is as it gets dispersed into these warmer regions. And so when we put that constraint in the model, the ocean doesn't get connected at all, as you might expect. And within 100 years, only 10% of these points have become connected. So then the question is, what if we allow them to evolve? And we use this same sort of trait diffusion approach, where we said species are going to produce offspring, and they're going to produce most of their offspring with the same traits, but some of them are going to have a slightly higher thermal optimum. Some of them have a slightly lower thermal optimum. And we initially said that 1% of each generation's offspring will go into its neighboring trait value. And when we do that, we get this sort of diffusion of traits, and we get slightly higher global connectance, but actually it's not very extensive at all. And if we go to a really, really high rate of trait diffusion of 10% of the offspring have mutant trait values, then in that case, we get maybe 85, 90% of the ocean connected within a decade. But these are extremely high rates of adaptation. So these different assumptions in the model, which we don't know which is the most realistic at all, but what we do know is that they have big impacts on the distribution of a population's descendants. So if we take an example species in the North Atlantic, in the neutral case, it can get to most of the global ocean. This is after 100 years, so it's reached full global extent, but it's mostly concentrated in its initial environment, but maybe has this sort of decaying abundance across the globe. And we see that these sites are clustering together fairly smoothly in one global province with maybe some environmental decay in community distance across those sites. In the case where we've got selection but no adaptation, the the distribution of the seed species is extremely restricted. We get a lot more structuring in the community. So different locations in the ocean have very distinct communities because they're restricted by their temperature. And as we allow adaptation to break down that restriction and dispersal, we move back to a sort of more a larger community clusters of larger geographic extent. And, we can try and compare this to the patterns that were shown in the Tara data. I'm not going to argue which one of these matches those best at all. I'm just going to conclude that this study sort of raises the point that everything is not, maybe everything is not everywhere because the environment is selecting, and there are these restrictions on dispersal because we have to cross these enormous environmental gradients. We can also note that full global dispersal of a population requires extremely fast rates of trait diffusion, and you could quite reasonably argue that 10% is very unrealistically fast rate of adaptation. What the model doesn't include is potentially other really important mechanisms like resting stages of phytoplankton or senescence, allowing them to avoid competition and just disperse more like passive traces. We could also think about ballasting in ship ballast waters, and even atmospheric transport being potentially important mechanisms for dispersal, but I'm not going to say anything about those. And it also raises the question of what if we represented these limits in biogeochemical models? How would species be able to track climate change if there were really strong constraints on their dispersal with isotherms moving towards the poles? Would that change the predictions of these models? Would we see species moving with the isotherms? Would we see them adapting to their changing local environment, or would we just see them dying out? So finally, I want to move to an individual-based model and look at this assumption about the environmental selection, and particularly looking at the competitive exclusion principle and some emergent patterns around that. So in this study, which is available as a preprint, you can email me if you want details of that. We found virtually unlimited genetic divergence can be supported in a simple system, even in the absence of niche separation. That's because of rapid evolution. So we used we looked at the observed high coexistence and functional redundancy in microbial data in the ocean, and we used an individual-based model to investigate that. So this idea of functional redundancy, here we have a functional map of some metabolic processes, and this is just the raw function. But if we look at what's actually doing that function, we can see that there's a lot of each of these grey circles is a different species. So there's lots of different species doing the same process, and this is functional redundancy. And if we just look at this particular example here, there's this idea, we've got three organisms doing exactly the same thing. But if we think about the competitive exclusion principle, this is Hardin's complete competitors cannot coexist, then you could argue that there must be some other niche differentiation that allows them to coexist, because if they were all identical but different species, one of them would outcompete the other one. And so here we can look at different mechanisms related to viral lysis, different substrate use. They argue lots of different things, grazer, resistance, so on and so forth. But the argument is because complete competitors cannot coexist, there must be some niche separation. One application of this idea was in a study by Nadav Kashtan et al., who looked at single-cell genomics of Prochlorococcus populations in a single location. And they found within individual ecotypes, so within cells that had previously been clustered together, they found massive amounts of genetic divergence. And each ecotype would include many highly divergent subpopulations with millions of years of genetic divergence between them. And they argued because they have such stable differentiation over these millions of years, there must be some niche separation. So this pattern suggests ancient stable niche partitioning. What I want to show with this model is that it is possible for these patterns of high divergence to emerge even without niche separation. So this is a clustering based on a single batch culture model with one environmental niche related to temperature. And we do get these patterns of stable, highly divergent subpopulations. So the model itself, I took from this paper by Beckman, Sherman, Henser, and it's just a batch culture model where phytoplankton are taking up nutrients, producing detritus, and you get this recycling. But interestingly, they represented the phytoplankton community in terms of individuals, and each individual grows in size until it reaches a doubling size. Then it splits into two individuals. And as it does so, it has imperfect replication of its traits. So there might be some change in its trait value of its descendants. And this is what allows evolution. So an individual will produce two children that have slightly different thermal optima. And then the one that has the thermal optima best matched to environment will have a better chance of reproduction in the next generation. And this is this very simple mechanism that allows evolution in the model. And so you can see when they ran that model in a stable environment, you get, they seeded with a load of different individuals across the thermal optimum from 8 to 22, but the environmental temperature is 15 degrees. So after about 1 to 2 years, you get the emergence of a single distribution of individuals with their thermal optima at or around the environmental temperature. So what's the actual diversity within this, we'll call it a population here, sitting on this 15-degree optimum? Well, when we added a mechanism for tracking the genetic divergence of these, or the lineage separation of these organisms, and we could track their lineages right back through the evolution of the model, we could find within that cluster, there's all these different subpopulations, and they've got up to 10,000 generations of divergence between them. So they're very distinct in their evolutionary history, although they have exactly the same, they're living in the same thermal niche. They have the same phenotypic characteristics. And when we looked at those lineages through time, we can see each of these lines, there's 20 lineages here from individuals sampled at the end of the simulation. And you can see they're showing this random walk around the thermal optimum. So there's a lot of lineages here, but if you go integrate over the long time period, they have a kind of equal fitness over that long time period, and that allows them to coexist as if they were neutral. They're not always the same, but they do manage to coexist. And this allows us to have this pattern of extensive lineage divergence in the model. And actually, when we look at this, we go back through time. These are the number of distinct lineages at the end of the simulation. And if we go back in time, we lose diversity. And this is through this basically a process of coalescence. This is the Wright-Fisher model, essentially. If you go back farther enough in time, you lose diversity because everything will go back to a single common ancestor. And this is a neutral model. But what this is showing is it's following this neutral model, but we have these selective processes in here. And the Wright-Fisher model says that if you have a strong population bottleneck, if the environmental conditions change, you're going to see a loss of diversity as everything is replaced by the descendants of the mutant species that seeds everything else. In this Beckman et al. Paper, they changed the environmental temperature suddenly at 1 year from 15 degrees to 20 degrees, and they saw this evolutionary response. And I'll just skip forward because I'm running out of time. What you'd expect in the classic view is you have a mutant that's better adapted to the new environment, and the descendants of that mutant take over the population and remove all diversity that was available before that change in the environmental conditions. This is called a hard selective sweep. And this is actually what we see with the coronavirus mutants through time. So here we've got the original composition of coronavirus in the environment. We have the Delta, the Beta variant coming in with slightly better, more competitive traits, and it pushes out all the other diversity. Again, with Delta and Omicron being this massive jump in fitness at a very rapid selective sweep. And this is arguing, this is a reason why we can't have diversity coexisting in these populations. But when we look at the model and we look at the lineages through time of this environmental change, if we change the temperature at 5 years from 15 to 20, it's really interesting that the diversity, the species that have adapted through this change in the environmental conditions were actually changing before the environmental conditions change. So what this is saying is the adaptive population has arisen from standing variability already available in the community. And that standing variability is available because of the extremely high population sizes in the system and the fast rates of evolution. So this means that I'm going to skip over this because I'm running out of time, but this is just a mechanism by which population bottlenecks can be weakened, and we can sustain this diversity through these environmental changes. So in terms of functional redundancy, the key takeaway from this work is that when you see three different species doing the same thing, it's not 100% necessary to invoke hidden niche dimensions to explain their coexistence. It could be that there's distinct lineages here that have converged to the same phenotypic conditions, but they're actually very distinct. So we have a lot of this neutral diversity. So when we see genetic diversity in environmental data, this need not imply functional diversity. And if we want to think about functional diversity, it's essential to identify those functional differences. And I think that's a really great thing about the work Adam was describing yesterday, is that it really focuses on these functional differences rather than just taxonomic differences, which we don't have tied to function very well. And in these very large populations that we see, they can sustain diversity for an extremely long time. And because I skipped over that point, I'll skip over that point. So just to conclude there, how I've used these studies to question these assumptions. So the population traits being able to adapt instead of the everything is everywhere assumption. This really alters the ecosystem recovery through disturbances. We found apparent barriers to dispersal, questioning whether everything really was everywhere, and also this high neutral taxonomic diversity can emerge in these fast evolving and highly abundant systems. So I'll leave you with this this illustration of Redfield's feedback and add this evolutionary process that I think we need to account for if we want to look at these systems as the complex adaptive systems, including environmental, ecological, and evolutionary feedbacks that they really are. So I'll wrap up.
Thank you, Ben, for a very thoughtful, very exciting set of work that you showed. Do we have questions? Adam?
Hi, Ben. That was a really wonderful talk. And I'm really impressed with how you link sort of ecology and evolution to sort of large scale properties of the ocean. My question is about dispersal. And I wonder if you can speculate a little bit about to what extent you think that your models can sort of capture the full extent of dispersal in the ocean. The reason why I'm asking that is that the population sizes in the ocean, right, are enormous, right? There's like 10 to the 28 cells. And I imagine that in sort of individual based models or which I think is what you're using, right? There has to be a lot fewer. At least it will be hard to sort of capture sort of extremely rare events, like sort of a crazy bug that sort of somehow makes its way through the deep ocean and then up into the surface. And so I was just wondering about, to what extent do you feel like that you can capture dispersal in these models? Are there any sort of uncertainties in whether or not sort of rare events can be captured in the models, or how that might affect sort of whether or not we can sort of assume sort of full dispersal potential versus sort of these restrictions that you suggest?
So I'll answer that in two ways. First of all, to say it's not at all constrained. There's so many uncertainties that I think the model results will be extremely sensitive to. But what I will say is that I think the way we did it did represent a bit of a step forward in that approach, because the way it's done before is either an Eulerian approach where you just mix a concentration or abundance in the way you do in global ocean models, and that potentially allows very low abundances to spread. And then there's the individual-based model, and the individual-based models, as you said, are limited by their the population sizes you represent. There's billions and billions of phytoplankton. We can't put those in an individual-based model. What we used was a population genetic model that, and I glazed over this very quickly, where you mix the population abundances, but then you draw the next generation from it based on like drawing from binomial or a multinomial. So you can actually put the real abundances in there. And as those abundances get down to low levels, from that statistical approach, you have a probability of extinction there. So if you have just 10 cells in an environment and you draw that from binomial, there's a very real extinction that they'll go down to zero. That probability is way too high in the individual-based models, and it's way too low in the Eulerian models. So we felt that this representation of it was slightly more realistic. But again, concluding that there are so many uncertainties that the model is very sensitive to, it's not fully constrained.
Great. Thank you. We have an online question. Jana, do you want to unmute and ask your question?
Yes. I really enjoyed listening to your presentation, Ben. If we have time for it, I have two questions. One should be rather short. I didn't understand why in the last part of your presentation, why your population is already increasing in T-op when there is no change in the selection temperature. That's the first question. And then the second one is a bit more maybe philosophical about you were describing in your introduction this feedback that adaptation can also have on the environment. And I was wondering how far we are from actually quantifying or somehow identifying this feedback and finding out how adaptation actually shapes the environment itself.
Okay, so the first one is effectively a matter of serendipity. This is a random walk that these organisms are going on in the trait space. So there's a slight change in thermal optimum at each generation. These ones are the long tail of the Gaussian distribution. And if the environment hadn't changed, they just would have died. But they just happened to be in the right place at the right time when the environment did change. And that allowed them to get through the change in the environment most effectively, and all these other ones that were lagging behind just died out. And the second question, I think you're asking sort of how close are we to representing the evolutionary feedback in global models?
Yeah, exactly.
I think there's technical ways to do it. The study with the dispersal, we looked at a population model because we didn't want to deal with those feedbacks. It's really tricky. We have a version of the cochlith for mass extinction paper in a 3D version of a very low resolution ocean model. It's technically possible to do it, but there's so many uncertainties around that. And I think what we're restricted to at the moment is doing sort of thought experiments, put this process in the model, just see what happens. Having it well constrained to the point that you would trust predictions from it, a very, very long way, I think.
I see. Thank you.
Thanks.
Ken, do you want to go ahead with your question?
Yeah, sure. Can you hear me?
Yep.
Yeah, well, thanks, Ben, for a really intriguing and tour de force, I would say, in the modeling here. I have a question to the middle part about the everything is everywhere, because you quoted Tom Finch's paper, and I took actually that paper really as an experimental validation that you can actually find everything everywhere, provided that it's sufficiently small, smaller than a millimeter. And I was thinking whether that a lot of that from your model comes down to this selection coefficient that you're imposing when things are advecting. So you have that Gaussian distribution there. And a lot of that will be determined by the tails of that one. What's happened there. I mean, you have exponential tails in the Gaussian, but if it was power law tails, which is very hard to distinguish, then I guess everything would be quite different, because then you would have something that could survive and cross these temperature barriers, wouldn't there? Oh, what are your thoughts on that?
So, I'm definitely not arguing against empirical data. I'm sort of trying to explore some of the mechanisms that might be driving it or have constraints on it. I still think it would, regardless of the tails of the thermal tolerance, I still think it's very difficult for a cold adapted species to outcompete a warm adapted species in the warm environment.
Yeah, absolutely. The question is not whether they're outcompeting, then the question is whether they are there.
So a lot of it, that's why I put that point in the conclusions of that section, is there's a lot of processes that, are not represented. It's a very, very simplistic model. We don't have senescence. We don't have aeolian transport, ballast waters, whatever. But I think what it's saying is the assumed mechanism of ocean transport, it actually seems very difficult for them to be able to do that. So maybe we need to look in more detail at what actually mechanisms do allow them to do it. If it, it may or may not be the case. It seems likely that it is the case, that they're very well dispersed, but how do they do it is perhaps the question.
Yeah, fair enough. Yeah, point taken. Thanks.
And then I guess, Shamit, did you want to go ahead?
Hi, do you hear me?
Yeah.
Hi, Ben. Thank you for the talk. I was wondering if there any difference in the outcome of the model if you include species evolution or versus you include the selection. Will be any difference in the output of the model? For example, if we want to understand how different organisms will respond to climate changes, and we want to understand which trait will be the most successful.
Can you be more specific about which process and model?
For example, if we want to understand which trait will be most successful if we want to understand what species will be the most successful under increasing temperature. So if we include in the model evolution or selection, will be any difference in the outcome, do you think?
It depends what level you resolve at. If we think about function, just sort of the broadest function, I don't think the high adaptive capacity suggests that function is very robust. So maybe there won't be big differences to the overall ecosystem function, unless you're thinking about really dramatic extinction events where everything is wiped out. If you look at the species level, I think it's completely unpredictable, and we have no chance of predicting what species is going to be dominant in a different environment in however many years, or even in the same year potentially. Perhaps, as always, in between, there's that interesting overlap between
the function is very robust, but maybe the taxonomic composition or the particular species composition is going to be more variable as a function of evolution, and that's perhaps where it could become unpredictable. So it's a very vague answer, but I think it really depends on the level of organization and whether it's functional diversity versus taxonomic diversity you're thinking about.
Hi, Ben, this is Jessica Liu. This is a very fascinating talk. As somebody who works on plankton modeling within Earth system models, I have always had the problem of thinking that, for example, the Epley curve for temperature responses to rates seems unrealistic under context of climate change. But in thinking about how to put in evolution into these models, or whether that's even possible, it seems like what we have here is kind of an initial condition problem. Like, where do we start these plankton populations from? And also how long we run these, like basically like kind of a pre-industrial simulations to get at a realistic ocean. And particularly in the second point, in how long we run these simulations, it seems like that's quite sensitive. Like the resultant populations are quite sensitive to that length of time. Do you have any thoughts on, basically how we can start to think about incorporating these ideas into our system models?
So in the original example of the Epley curve, I take your point that it does seem, it's a really crude way of incorporating what the data shows is a very broad array of different thermal functional responses. You could take the view that the evolution capacity is extremely high, and actually, no matter what we do, evolution is going to fill out that space under the Epley curve and give rise to that. So perhaps by exploring that, we actually give sort of more support to the that crude parameterization. That actually does make a lot of sense. So that's the sort of the robust functional capacity of the ecosystem. But then as you get to more fine scale questions like biodiversity, how is biodiversity going to change with global warming? Perhaps it's more important to investigate those feedbacks and include them in your models. I don't have the answers to questions like that. But I think it's worth exploring it. I think there's potential for those changes, those evolution changes, to impact the trajectory of the ecosystem under global warming. But it's dependent on this balance of whether taxonomic diversity is important or whether the functional response is really robust because of high evolutionary adaptive capacity. Does that answer your question?
Yeah, she says yes. Thanks very much, Ben. I actually have a bunch of questions. I'm sure other people do. Maybe we can take them up in the Slack. But in the interest of time, we should probably move on.
Thanks so much.
And our next speaker is Alana Smith.